talk about some mathematical modeling that are made specific to patients to improve the drug treatment.
Okay, thank you for this very nice introduction. I'm very grateful for the opportunity to talk here today. The project that I'm going to present today is a close collaboration with Theranostics, which is a company in Berlin which primarily works on precision medicine. And one of the key challenges in precision medicine is to design individualized treatments for every patient. And if you start out with a patient A who has, say, cancer, and you give that person drug A, and she directly responds to that, then you're very happy and you move on. But if you go to person B and you give him drug A and he doesn't respond, and you give him drug B and he still doesn't respond, you might, if he's still alive, you might give him drug C and he will finally respond. So this is really not the ideal case for the patient, and is also very cost intensive. So you ideally want to treat every patient at the right drug, with the right drug at the right dose. And for the purpose of this talk, I'll make the problem a little bit easier and derive a method on how we can treat every cancer cell line with the right drug at the right dose. And there are large online databases which characterize cell lines and patients in terms of their molecular profiles, so transcriptomic and sequencing data, and also in terms of their response to several different drugs. And what you would typically do is you use a statistical model where you have basically a linear sum, which is where every summed is the multiplication of a coefficient with a feature, which in this case would be sum of the molecular data that you have for that patient or that cell line. And you can then match that to the drug response data via some nonlinear function g. Now, our approach is that we additionally integrate all that biological knowledge that exists about the underlying system, and that we recast these properties into a differential equation, where the right-hand side is defined by this function f, and the function f defines the temporal evolution of all the concentrations of all the different molecular species in our model. And we can also take into account the molecular profiles x, and we again also have to train the parameters of that right-hand side of the differential equation based on some training data set. And the key advantage of that approach is that it really allows the systematic integration of knowledge as well as data, and it also allows the prediction of latent variables. So latent variables are species that were not experimentally measured, but as you model all the different concentration of molecular species in your system, you also gain access to everything that can be predicted from those species. Now, for this particular project, we looked at EGFR signaling, so we had 4 different receptor tyrosine kinases, and we looked at the downstream signaling via RAS and ACT down to several different transcription factors, which we can then link to cell proliferation. And this also includes the MYC transcription factor that Jana already talked about in the first talk of this session. And the model in total describes around 2,600 reactions and describes 1,200 different molecular species, and all the reactions are defined by 4,000 different kinetic parameters. So this is a very detailed model and a quite large-scale model. So this model was developed by Theranostics, our collaboration partners, and if you compare this to really large-scale models of cancer, such as the Atlas of Cancer Signaling Networks or the RECON 2.2, which is a metabolic model, it's on a similar scale, but has much more detail in what it actually describes. And now, in order to make this specific to individual cell lines, what we assume is that you have the biochemical reactions, which define a biotemplate signaling network of reactions that are realizable in every specific cell line, and that the sequence information as well as the gene expression levels then provide you with a cell line specific realization of the network. In the following I shortly explain this, how we realize this in our model. So here we have the example of two different cell lines. The first cell line has a wild-type KRAS, and here the gene expression level defines the protein synthesis rate of KRAS. And then you have subsequently all the different biochemical reactions. So you have the binding to GDP, then you have the upstream activation from EGFR, and then you have the downstream binding to PIK3CA, which then induces all the downstream signaling. Now, if you go to the second cell line that has a heterozygous mutation in KRAS, you see that it maybe has a little bit lower gene expression level, so also the protein synthesis rate for KRAS will be lower, but all the downstream signaling, so all the reactions that take place afterwards, will have the same kinetic rates as for the first cell line. But you also have a mutated KRAS version, and there you also have maybe a different different synthesis rate due to the difference in the gene expression level, and there you also have different translocation and phosphorylation rates, and also the mechanism of action is different here. So for example, for this G12 mutation, we assume that KRAS will be constitutively active without upstream activation from EGFR. And again, these rates will be, this is different from the wild-type mutant, but it will be all the same for if we go to a different cell line that has the same mutation. Now, in order to make these models really predictive, you have to parameterize these models, and so the parameterization is performed on the kinetic rates, so everything that is not synthesis is not defined by the molecular profiles that we recorded. And the parameterization that we do is typically done via optimization. So you define an objective function which gives you the distance between the simulation as well as the experimental data, and that defines a high-dimensional objective function landscape, which can have multiple local minima that you want to find, and you typically want to find the global minimum. So you have to do repeated optimization in order to find the global minimum. And all the optimization methods are implemented in the MATLAB toolbox Pesto, which is available online. Now, the key challenge here is that for these large-scale models, the gradient computation that you need for the local optimization can be computationally very expensive. So even if your simulation time is relatively low, so say 10 seconds, you need to simulate your model for all the experimental conditions that you consider, and you need to consider a large number of experimental conditions in order to parameterize a large model. Then this scales linearly with the number of experimental conditions, and additionally, if you want to compute the gradient, this again scales linearly with the number of parameters that you have, so you end up with a total computation time of over 6 years for every gradient evaluation. And now, you're not done with one single gradient evaluation, so you have to do this multiple times, so this is absolutely not feasible with state-of-the-art methods. And of course, you can now wait for 50 years until you have faster computers, but that will structurally not solve the problem. As soon as you go to a bigger model, you will have the same problem again. So you really need to structurally change the approach that you take here. And for this purpose, we used a joint sensitivity analysis. So finite differences and forward sensitivities are the state-of-the-art methods that basically everybody uses. And as I previously said, those methods scale linearly with respect to the parameters. And compared to that, a joint sensitivity scales basically constant with respect to the parameters. So you really achieve a scalable alternative that makes this the parameter estimation tractable. And we published this method early on this year. Now, if you combine that with specialized solvers that exploit the sparsity structure in your ordinary differential equation system, then you can achieve 30,000 37,000-fold decrease of the computation time. And if you add on top of that parallelization, you go to 240,000-fold. So you go down from 6 years per gradient evaluation to something like a couple of minutes that you require for every gradient evaluation. And this basically makes the whole approach feasible. So if you can do that, the question is can the model actually explain the data that we have? And for this purpose, we looked at growth assays from the Cancer Cell Line Encyclopedia, where they recorded the relative proliferation with respect to different drug concentration for various drugs as well as various cell lines. And there we have on the top, we have 6 different cell lines. So we have resistant cell lines, we have weak responders, and we have strong responders. And the model can describe relatively well the curves that you see in the experimental data. Now you also, I hope you can appreciate that we have performed 5-fold cross-validation, and you see the 4 different lines in every plot correspond to the different realizations, and you actually find that we can really reproducibly find a good fit of the experimental data. And in order to give you the bigger picture, what happens with all the different cell lines and all the different drugs, I look at the correlation between the data and the model simulation at the highest drug concentration, and there we find that we have a correlation of over 0.8%, over 0.8, which is quite good. And if you look at the correlation for different drugs, you find that for certain drugs you achieve a much higher, a little bit higher correlations. For example, for the selumetinib on the bottom right, you see that we achieve a 0.9 correlation, and for other drugs the correlation is not as good, which tells us that we are maybe not arbitrarily overfitting the data, and that this could be an initial starting point for further model refinement, as the understanding of the action and the targets of the different drugs in the model is not completely understood. Now our starting goal of the whole project was to really also predict the drug response of new cell lines, and for this purpose we classified all cell lines in responder and non-responder cell lines based on their response at the highest drug concentration. And we again looked at, performed a 5-fold cross-validation with cell lines from 5 different tissues, and also considered an additional data set where we had additional cell lines from 4 different tissues that were previously not considered. And we compared our mechanistic approach with various statistical approaches, and we found that on both data sets the mechanistic approach significantly outperforms all the investigated statistical approaches. And here I really want to stress that all the methods only used the training set for the parameterization, so the estimation of model parameters as well as of hyperparameters, and the test set was never considered for the training of the model. Now what I already hinted at in the beginning of my talk is the prediction of latent variables, so things that were not experimentally measured. And for this purpose we looked at combination treatments, and there we again used the single treatment data from the Cancer Cell Line Encyclopedia, and we also had data for combination treatments, and there we found that our model achieves a correlation of around 0.25 to 0.3, which is not particularly good. But if you compare this to the correlation between the data itself as well as the correlation for the single drug predictions, we find that the correlation is not much higher, and that we think that the problem is actually that there is a disagreement between the two data sets, and that the poor correlation is actually not due to a shortcoming in our model, at least to the extent that we can assess it. And with this, I want to come to the end of my presentation, and to sum everything up, what I wanted to show you today is that mechanistic modeling really allows the systematic integration of prior knowledge as well as of multi-omic data sets. So for this particular application, we looked at sequencing data as well as transcriptomic and phenomic data, but we're also working on proteomic and phosphoproteomic data now, and that the parameterization of these large-scale models is really only possible if you use a combination of adjoint sensitivities, exploit the sparsity of the system, and use parallelization. And that the parameterized mechanistic model that we derive in the end is really able to outperform statistical approaches in terms of prediction accuracy, and that we can enable predictions for latent variables such as combination treatments, which is not possible with standard statistical methods. And with this, I would like to thank my collaboration partners and my supervisor, especially Jan Hasenauer in the group of Data-Driven Computational Modeling, and you for your attention, and I'm now happy to take questions.
Hi there, great talk. One question about the parameters. So it's possible to have models that have a large number of species but a very small number of parameters, or for example, a simple polymerization reaction. So in the model, you mentioned that there were 1,200 species and 4,100 kinetic parameters. Were those all for independent reactions, or were there instances of reactions involving sort of trivially distinct species with the same reaction mechanism that would have the same parameters?
I'm not sure whether I got the question. So we have reactions that have multiple kinetic parameters, and you also have basically the same reactions for the mutated variants, sometimes for the mutated variants of a specific protein, but we do not exploit this structure in the model.
Mm-hmm. I was thinking along the lines of complex formation and signaling, for example, where two proteins, the binding mechanism is local, and that has a certain kinetic rate, but depending on the post-translational modifications or the other things that are bound to those things, you can get distinct reactions in an ODE model, but the binding event between those two proteins may be kinetically independent of that additional context. So you could actually use the same rate parameter for those two different reactions. Is that?
No, we don't exploit any of that structure at the moment.
Thank you very much for this nice talk. You say at the beginning that you combine the treatment between the drugs, but did you try also to predict the timing at which this drug can be combined, if at the same time or a different timing?
No, at the moment we do not consider this. So I think this is a big topic, especially in terms of drug resistance. But this will, I would guess that this would at least require the inclusion of gene regulation in the model as well, and that is currently not yet done.
Thank you.
Thank you for your talk. So if I understand it correctly, you had about 4,000 parameters and 5,500 data points. So to me that still sounds like an unfavorable ratio. So this adjoint sensitivity analysis, does that perhaps bring you in a position, as the previous question basically alluded to, that it actually effectively reduces the dimensionality of your problem?
It does not. The problem, I totally agree that the ratio of data points with respect to the parameters is not ideal here. But in this project, we're really not that interested in the individual parameters, but we're more interested in the predictions. And there we find that we can really make robust predictions. So we do not care so much about the parameters at the moment. But ideally, of course, you want to gain really a mechanistic understanding of what is happening. And I think for that, we will need proteomic and phosphoproteomic data. And then you have really many more data points. And I think then you can also reliably estimate all the parameters.
But shouldn't one be skeptical of a model where you don't trust the parameters, but it predicts kind of accurately?
Yes. So that's why we want to work with proteomic and phosphoproteomic data now.
I had another question, and that's you correlated the responses at the highest concentrations, which was 8 micromolar, if I saw it correctly. So don't most of the cell lines die at that concentration?
So if you look at the data from the Cancer Cell Line Encyclopedia, it's not the case that, so we definitely have cell lines that do not respond to the drugs at that concentration.
Maybe it's simpler if you just look at lower concentrations. How does the correlation change then?
The problem at lower correlations is that you get much better correlations because you have many cell lines that do not respond at all, and that is relatively easy to predict.
Okay, thank you.
So how do you use your model in the context of your original question of identifying the responder, which patient will respond better? What's your response variable? Do you link this to tumor volume or any kind of clinical?
So in the application here, we didn't really take the step yet to go to clinical applications. Everything is done in cell lines, and evidently going to really to the patient level and making predictions there is what we want to do, achieve in the end. But for that, the model has to probably be combined with agent-based models and evolution-based models, and you also have to take into account cell-to-cell communications. So there's so many different things that still need to be done, and there are so many opportunities to work on that, but we're just not there yet.
And just a similar question to the previous question. Are you fitting all the parameters, or you fit a subset of the parameters?
We fit all the parameters at once.
Okay, thank you. I think we have to move on.